enzyme called Aldolase, which is a lysase that cleaves fructose 1,6-bisphosphate into dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or GAP. The product will be broken down later by other enzymes, and the enzyme plays an important role in glycolysis and gluconeogenesis. The aldolase is a homotetramer made up of alpha helices and beta sheets, which leave space, here you can see at the active sites, for binding to a substrate. Different forms of aldolase exist in humans with different binding site specificities. The catalytic site, however, is highly conserved, and the enzyme, as you can see because of the 4 subunits, can bind 4 different substrates at once. Here you can see the mechanism, or the general reaction which occurs. The cleavage occurs between carbon 3 and 4 and results in the formation of 2 3-carbon chains, both with a phosphate group. It is a condensation reaction, meaning it releases water, and some isozymes of aldolase use a cofactor, although this one does not. The binding site interaction is a result of salt bridges that occur between the backbone amino group on serine 271 and glycine 272 with the 1-phosphate, as well as the formation of salt bridges between positive lysine 41 and arginine 42 and 303 residues with the negative oxygens on the 6-phosphate. This binding site interaction holds fructose 1,6-bisphosphate into the enzyme while the catalytic reaction occurs. Fructose 1,6-bisphosphate must be in the open chain form to be able to bind, and a base is required to open it from a ring into this long chain. Once the substrate is bound to the enzyme, lysine 229, which has an amine on the side chain, and it reacts with the ketone on carbon 2 to form a protonated imine, while the oxygen will then grab onto hydrogens to form water, as you can see here. The formation of the imine involves a covalent interaction, which makes this a covalent catalysis. Aspartate 33, which is deprotonated and negatively charged, deprotonates the hydroxyl on carbon 4 and forms the aldehyde on GAP. GAP is no longer bound to the enzyme and is able to leave the active site. Aspartate 33 can then be protonated, which provides the hydrogen required to shift the electrons back onto the lysine and back onto the DHAP, and causes the carbonyl to form back on carbon 2, which completes the mechanism. The formation of the carbonyl means that DHAP is no longer covalently bound to the enzyme and allows it to leave the active site. Here you can see those 2 residues involved, and the main takeaways are that the substrate must be modified before it is able to bind. The enzymes can bind 4 substrates. The binding site and catalytic site utilize different residues to interact with the substrate, and that this reaction is important for glycolysis and gluconeogenesis.